I just want to give a shout out to Mike for inviting me to tell you all the story of this pretty amazing species today. So just an intro about myself. I've been working at the DEC for the past 5 years and about the last 4 on bat conservation. I'll tell you a little bit about what we started finding on Long Island, which really spurred my research interest, and I decided to go back to school, working in the Ecology and Evolutionary Bio department at UAlbany. So although my title here is Bat Conservation on Long Island, pretty much 100% of that is really surrounded by the northern long-eared bat here on the island. So that's really our focus species here today, and I'll be telling you about why we're really experiencing some truly unique situations here. So if you aren't familiar with the other species of bats that are found on the island, I'll just give you a little bit of an overview. So these 3 are our tree bats or migratory species. So generally it's thought that they're only here in the spring and the summer, and then they migrate further south in August and early September. So you have a silver-haired bat, hoary bat, and the red bat. They're all pretty cute and colorful and furry. And not a lot is really known about them. There's a lot more research going into their migratory patterns as of lately, and it doesn't seem that it's as straightforward as most bird migration is. So I have some evidence to show that I think even red bats stick around during the winter. So there's a lot more research to be done there. In terms of your species that hibernate here in New York and stick around year-round, you have a big brown bat, your most common species at this point in time, and also seems to be the least affected by white nose syndrome. And if you aren't familiar with that or what it is or what's going on, I'll tell you a little bit more about that in a few slides, but just know that it's not good for bat populations. But this species seems to be doing well, and many records all throughout Long Island, both Nassau, Suffolk County, they do tend to hibernate or overwinter in people's homes. So it can be really hard to track their populations by just going into a cave or a mine and doing a population count. But we do get good records using acoustic surveys for this species. Little brown bats, probably the most, they used to be the most common in New York and the ones you hear the most about in terms of white nose syndrome. Also plenty of records of this species throughout Long Island. They do seem to be okay with using human structures as well during the summer when they form maternity colonies. But it's thought that they do fly back to mainland New York or Connecticut and overwinter in a cave or a mine system. And the tricolored bat, very rare species overall in New York, and there are just a few records, I think back from the late 1800s of the species on the island. We don't really ever catch them or detect them acoustically, but they are pretty rare. Also one of the species that is the most affected by white nose syndrome. We've seen population declines of about 98% in New York alone, and that's really been mirrored throughout the rest of their range further south too. So onto the northern long-eared bat, Myotis septentrionalis. They go by many other names. You'll probably see me refer to them as myci, just given their scientific name as a short note, or northerns, or seps for the septentrionalis. So I'll try to just refer to them as northerns. They were formerly widely distributed throughout the state. You could pretty much catch them anywhere or find them in any hibernacula. They were federally listed as threatened in April of 2015, solely due to the population declines from white nose. So here in New York, we've seen declines up to 99%, except for Long Island. So there's been a really unique situation going on down here, and I'll tell you more about that. They can be pretty difficult to monitor with both of our typical population monitoring methods. So we do a lot of hibernacula surveys in the winter, just going into caves and mines, counting all species and individuals of those species. But they're cryptic in the sense that they do like to hide up in little cracks and crevices. They don't form big clusters, so it can be hard to find them if you're doing a survey, even if they are there. Acoustically, they are often called a whisper bat, so they have a much lower pitch to their call that they echolocate. So they can be hard to detect, but as we've seen on Long Island, it's actually pretty easy to detect them down here. So they get their name, they do have slightly longer ears than your little brown bat. Otherwise, they look pretty similar. But what's really unique is they have a really long tragus, so this little part in the middle of their ear. And that's really one of the only ways you can tell them apart if you have both species in your hand. So just to give you an overview of what their entire range looks like, it's this green outline here. So they do extend up into Canada and into the Midwest, a little bit in the South as well. This is an image from the Fish and Wildlife Service. So because they gave them a threatened listing, what they have done is implement this 4D rule. So there's essentially less restrictions than an endangered listing on the species. And this was really due to advocacy from the forestry industry, which was pretty much worried if we listed them as endangered, it would mean more restrictions on tree cutting and forestry overall. So with the 4D rule, there are some restrictions. You can't just go into a hibernacula and disturb them or kill them or bother them in any sort of way because they are very vulnerable during the hibernation season. In terms of tree cutting on the actual landscape, if we have a known occurrence, so maybe there's a known roost tree where there's a maternity colony, you're not allowed to cut trees within 150 feet of that tree. And it's recommended that you do this outside of the pup rearing season, so the middle of the summer when they're really vulnerable and they're raising their young. They usually leave the young in the tree until they're old enough to fly on their own. New York has taken a slightly more strict approach in what we recommend, so we have sort of extended those buffers. And we have some additional recommendations. If you can cut your trees in the winter, that's just the best option because you're not going to worry about disturbing roosting bats or a maternity colony. And Kevin Jennings is our regional biologist that deals with this down on Long Island, and he's done a really great job of getting the word out there and providing those recommendations for people that do need to cut trees. So. What is white nose syndrome? It is a disease affecting hibernating bats throughout North America. It's been responsible for unprecedented mortality. I think the estimate was roughly 6 million bats have died from the disease, but that was back in like 2011. Probably an underestimate at that point, so it's definitely millions more, I would probably say. It comes from this, the name comes from what we typically saw in the early years, so this growth of the fungus on any exposed skin membranes, so covering face, wings, ears, and it's the actual white fuzzy fungus growing on them. The culprit here is Pseudogymnastus destructans, or short is just PD, because that's a long name. This is what it actually looks like, the fungus when it's growing. And this is a histopathology view of the wing membrane. So what the fungus actually does is it grows into their exposed skin. It causes a lot of disruptions. And the main way that it actually kills the bat, so we think, is because it's causing them to wake up more naturally than they normally do. So bats that hibernate, they don't just hibernate for the winter on a normal season. They actually wake up roughly every 3 to 4 weeks. Not entirely sure why they wake up, but probably just to get a drink of water, maybe boost their immune system, and then they go back into a state of torpor. So that's natural, but a bat infected with the fungus will wake up more naturally than it normally would. This causes them to burn up all those fat reserves that they built up to survive the winter. They end up coming out on the landscape and trying to feed, and in the middle of the winter, there's not many insects out for them to feed on, and they end up dying or getting eaten by a predator or something like that. So that's the mechanism of how the fungus is actually causing mortality. And there's some interesting seasonal dynamics to the disease, which I will show you in one more slide. But just to give you an overview of the spread of the fungus so far. So it actually originated right here in New York, up in Albany and Schoharie counties in 2006-2007 winter. Since then has spread quite far, really following sort of the topography and natural cave systems throughout eastern North America here, up into Canada. And unfortunately, in 2016, jumped all the way over to Washington State. What we do know at this point is that it's all clonal dispersal of the fungus, so it's all just one strain being spread throughout the country at this point, which is a good thing, we think. If the fungus starts reproducing sexually, there could be other strains that might affect certain species that are less affected right now. So it's currently in 31 states and 5 Canadian provinces. So just over a decade has really spread quite far. The method of spread is really just bat-to-bat transmission. So bats will go into a site, pick up the fungus in the cave or mine environment. A lot of them like to cluster together in big clusters, so then they're spreading it around to other individuals. And then in the fall time, that's their mating season. They come in contact with other individuals and keep spreading it from site to site. So especially somewhere like New York, it's virtually in every site that we look in, and it just really persists indefinitely in the cave environment. It's a very specific fungus, so it only really grows at a certain temperature and a certain humidity level, and that just so happens to be the temperature and humidity conditions of most of our hibernation sites. So it's just kind of the perfect storm for infecting bats. It likely came from Europe, so there's been a lot of research done into trying to identify exactly where this strain came from that we have in North America. The problem is in Europe, the strains are very diverse, so it's going to be probably difficult to pinpoint exactly where it came from, but it seems the closest related strains are Western Europe, but it's virtually everywhere people have looked so far, in Europe, in Asia. People are starting to look in the Middle East now as well, but it's just everywhere. The interesting thing is the bats over there are not experiencing mortality, so no widespread death. You do sometimes see individuals with the fungus growing on them, but it just doesn't seem to bother them. So this sort of indicates, or people have suggested, that there's been a coevolution between the pathogen and the host. So it could be a positive outlook for our species. Maybe eventually they will develop this tolerance, but it might be too late for those that are already experiencing such drastic population declines. So hopefully you can see this. I'll explain, but this is some great work that was done out of UC Santa Cruz, just looking at that seasonality of the disease and how it spreads. So Kate Langwig, too far. She went into a bunch of sites, swabbed a bunch of bats, and you can run the swabs through PCR and detect the fungus. Not only detect it, but see how much each individual is carrying. So she did this in the fall swarm period. So again, that's when they're mating. They're coming in contact with a lot of other individuals. They usually go in and out of hibernation sites, so they're picking up the fungus there and spreading it around. So you see most species pick it up at this point. Once they enter into hibernation, this is time on the x-axis, and then just estimated prevalence or proportion of individuals infected. It skyrockets pretty high. And then if individuals can survive into the summer when they emerge and go on the landscape and join maternity colonies, they can essentially cure themselves of the fungus, or at least rid themselves of it until they go back into a site again in the fall. This is a similar figure just showing loads, so how much fungus each species is actually carrying. And the real takeaway here is just there's a lot of differences between species. So the orange color here is the northern long-eared bat, and you can see they are one of the most heavily affected species. So almost once they enter into hibernation, just virtually the whole population is covered pretty quickly, and they carry some of the highest fungal loads as well. So probably indicating why they are so heavily affected by the disease. So what about northerns on Long Island? Some of the first records come from, or at least published records come from the mammals of Long Island that you may have read or seen in your research from the museum. So the first record was in 1933, and this was actually an individual that was found in somebody's house. I forget where, but I think it was local, close to where we are here today. So remember that for later, that they found this individual in a house as well. The author of this study went out and did some misnetting to just see, what species are out there at the time. He was taking specimens back to the museum as well to study for morphological characteristics and things like that. And he went out netting way out on the South Fork in Hither Hills State Park at Fresh Pond. So there was like a freshwater pond on the property. I think he netted over, it was maybe like 4 nights over 2 years, and caught 37 individuals, just northern long-eared bats. No other species of bat at the time was caught. So this is pretty impressive, and it kind of indicates that at least at this point in time, they were the most prevalent species, at least this far east on the island, which is pretty cool. Our only other records that we really have from historical records are the submissions that we get to the Department of Health rabies lab. So if somebody has a bat in their house and they're worried if they were exposed to rabies or if the bat was rabid, they'll submit it and it gets tested. So historically, we would have roughly one submission per year from Suffolk County. So what about now? White nose was first found right here at Brookhaven National Lab in 2011. So Tim saw a bat out on the landscape in the middle of the winter, tested positive for white nose, so that was our first detection of the fungus on the island. How that probably happened is, we've always assumed that these cave-dwelling bats are just going to fly up to upstate New York or Connecticut to a hibernation site to spend the winter, and then come back down here in the summer. So possibly how it got brought down here. But this individual was found in the middle of the winter, which might indicate that it's spending the winter on the island. Mike Fishman, who is now working with ERM as a consultant, did a bunch of netting here on the property as well as at Wertheim National Wildlife Refuge. And he also caught a large number of individuals across these two sites. Usually we record nights and net nights, so that's how many nets you put out on the landscape, multiplied by how many nights you actually were out there. So this is actually really high numbers, and also really indicates how prevalent they still are, or so we think. After white nose, we know it's here. And we also see an increase in submissions to the Department of Health. This is largely due to an initiative they started in 2007. I think it was called the bat in the bedroom. If you have a bat in your bedroom and you're not even sure if you came in contact with it or if your child came in contact with it or a pet, you should just submit it to be tested and not assume that, I didn't feel a bite or I didn't experience one or I was sleeping. So that's probably why we have increased submissions, but it is still an indicator that they're persisting here. So DEC decided to start doing some mist netting down here because we saw these other results that people were having with this species. And just for comparison, I included the little brown bat here, and this is all just netting that occurred all in Suffolk County over the past few years. Haven't caught a little brown bat in a long time, but we have really good success catching northern long-eared bats here. This is just a proportion of net nights here, so that's our northerns. And what's interesting is this number is really similar to capture rates that we found on mainland New York before white nose. So also not great records pre-white nose, even on mainland New York, but what we do have, we're seeing really similar numbers to what we found there. So this is another really good indication that perhaps the population here is persisting for some reason. So what else is different about Long Island? I'm sure you all probably have some feelings and some thoughts about what might be different here compared to upstate New York. Just looking at land cover, a lot of diversity here. We have different levels of development in the different red shades, different forest types in the green, agriculture, and of course a lot of wetland systems as well. The further east you go, the more forested patches you tend to see. The other thing that's obvious is the topography. Chris showed a lot of good maps showing elevation as well as the geological evolution of this area. There's no natural cave systems on the island, so no natural hibernacula that we would expect to find these species overwintering in. So that really gave us an indication that they must be using something else that we would find here. And that's probably a lot of houses. So what I am proposing and looking at through my research, or what I think is happening, is the individuals in these coastal populations on islands are behaving differently. So they have some sort of just behavioral differences, unique behaviors for hibernation that are allowing them to persist despite infection with the disease. So I've come up with a bunch of questions that I'm trying to explore, and I'll talk to you about each one and how I'm going about them. The first one is really what's the population trend? So we know that we have these numbers here, but is this just indicative that we always had a larger population and maybe the decline is just slower? Maybe we're just not experiencing the decline that we see upstate just yet. Maybe there's not enough intermixing between mainland and the island. So we really just want to know what the trend is, which is going to mean we need to keep monitoring over time and seeing if these numbers persist. I'm also interested, year-round, obviously, activity looking at the winter and fall seasons, which are really understudied. What's their hibernation behavior? Where are they hibernating down here? What kind of structures are they using? How can we characterize these sites so that we can find more of them? What's the microclimate like? Most bat species have slight differences in the microclimate where they will hibernate. Northerns are a little bit more of a generalist than some others, but they still have a range of a temperature and humidity. They like really high humidities, roughly 40 to 50 degrees Fahrenheit conditions, and it has to be pretty stable, so it can't be like fluctuating every day. What's the exposure here to the fungus? We have individuals that have tested positive, but is it really prevalent within the population? And are they carrying really high fungal loads like they do upstate or throughout the rest of their range? We need to know that as well to understand how they're dealing with this. And I'm also curious a little bit about genetics. I am not a geneticist, but I have some great collaborators that are. So I'll talk about that, but I've been taking some samples to assess, maybe these island populations are pretty isolated here, and they really don't intermix with the rest of, New England and New York. So maybe they've been isolated for a long period of time. Maybe they have some genetic differences that are allowing them to, exist and persist with the fungus. So just for my first question, if we tried to address this with mist netting, it would take a lot of effort. So it takes a lot of time not only to set up, but just waiting for the bats to come, and especially in these, these lower densities, it would be a lot of effort if we were trying to do this just through mist netting. So what we do instead is use acoustic surveys. You can have a little acoustic detector that you can put out on the landscape, leave it out as long as you want. And it'll record sunset to sunrise and pick up anything that you set it to. So certain frequencies, really just focusing in on those bat ranges that they echolocate at. So 2015, 2016, we did a pilot study just to see how feasible this would be on Long Island. And this was really opportunistic in terms of the locations where we surveyed. So we had some regional technicians at DEC here that went out and put the detectors out for me. They were mostly doing this at easily accessible properties that were DEC properties, so state parks or other town lands that we had approval for. So again, we didn't really have great methods at this point. We were just putting the detectors out, leaving them out for a week on end just to see what we would get. And we had 130 detector nights, which means, one detector, you leave it out for a night. It's a detector night, so combine that all together, 130 detector nights across these 13 sites. Where did we actually find them are these areas in the red here. They were present at 9 sites out of the 13 that we looked at, and our detection rate was about 64%. So I don't know if you are familiar at all with occupancy modeling, which is what we decided to do with all this data. But essentially, you want to have a detection rate that's around 60% is pretty good for this. If your detection rate is 100%, it's not really worth it to even be modeling, or if it's too low, you're not going to have great data. And the idea with occupancy modeling is to pick your sites, go out over a set period of time, and once you detect that species, you can stop looking at that area, consider it occupied. So this just shows nights, so how many nights we left a detector out versus the probability of detection. Just leaving one detector out for one night gave us that 64% detection rate, which is pretty high. We found that if we left detectors out for 3 nights, we were like 95% sure that we were going to detect them if they were actually there. So this is really good for us. You can go out and leave detectors for 3 nights and be pretty confident that you will detect them and find them if they are actually there. So we wanted to look at what was influencing that detection rate that we were finding. So we looked at land cover, and we were looking at the proportion of surrounded habitat to our detection sites, and we did a 1.5-mile buffer. And this is because this is the buffer that the federal government and the state government kind of uses for a foraging area or a home range during the summer. There isn't a lot of data to back that up, that that's the best number for this species. So some of our work is looking into that as well, but it's what we decided to use just for this initial research. So land cover type here and proportion of surrounding habitat within that buffer. We found kind of what we expected. In your areas in the yellow here are where we actually detected them, and this red dark orange is where we didn't. So your forested areas were really where we had the highest detection rates. Makes sense, this is a forest-dwelling species. They're often called gleaners. They like to hawk insects off vegetation, whereas some of your bigger bats might travel down a roadway or forage over an agricultural area. They tend to forage more in denser forested areas or along trails. Obviously, development had a really significant negative impact. The more development you had surrounding your detection area, less of a chance that they were going to be present. So this is just showing when we actually did the modeling, development was the only thing that really came out significant. So once your surrounding habitat was about 60% developed, then your detection rate significantly dropped off. And that makes sense. Like the purple are where we found them and the black is where we didn't. Obviously, you can see it's pretty heavily developed there. But this was really a broad scale view, and I was interested in looking at things on more of a fine scale. So what I've done is develop a new sampling design that we started last year, and we're continuing for the next few years. So a really more intensive design for our acoustics. So I created this grid system, 1 by 1 kilometers, that covers all of Suffolk County. Decided to limit this to Suffolk County just because of the rate of development that we see in Nassau County. And then I wanted to look at just divide it by canopy cover. Typically, you would expect to see them, most present in sort of the middle or higher canopy cover levels. But I think that might not be the case. I think they might be using even smaller little patches of habitat that we find. So I wanted to split things up this way. And I have 50 study sites this time. So 50 the little black squares that are covered in are just the random sites that I selected, and I selected an equal number for each canopy cover level. Within each of these little grid squares, I have 3 detectors that I put out. So I'm sampling a small area, but I'm making sure that if they are there, I'm going to probably detect them on one of those detectors. And if I do, then I can consider that square occupied. And the stars are just showing you where we found them previously. So this is what bat calls look like when you look at them on the computer. And I spend endless hours just going through files and looking at all these little bat squiggly calls. You start to go crazy after a little while, but it is fun. So each species sends out a different echolocation signature, and you can tell them apart basically by the frequency and the slope of the actual call that they're emitting. It's pretty incredible. Within they basically send out 3 signals every 300 milliseconds. So a lot of signals are coming out constantly. It's great because we can record them with these little detectors here. Things to think about when you put detectors out are really just placement. Obviously, you want to put them in places where you're going to get good quality calls, and also the species might be there. So you don't want to have it surrounded by too much vegetation, because when they are out there sending out their little echolocation signals, they're getting signals back, and it's bouncing off everything, and that's how they navigate, and they find bugs to eat. No, that's fine. Yes. Yeah, no, these are all the 9 species that we find across New York. Yeah, so you have your low frequency ones, and then up here are your, these are your Myotis species. So you can see that the 3 Myotis, is it tricolored, who just has a really high frequency, but it's a low slope. The 3 Myotis are, I don't know, they look more close to each other than many of the other calls do, so it can be really hard to differentiate between the 3. But luckily, northerns, this is a northern call here, are the steepest and the highest ones. And somewhere like Long Island, we don't have Indiana bats, another Myotis species, and we have really low populations of little brown bats. So when you get a call that looks like this, you can be pretty certain it's a northern long-eared bat. So that's why it also works really well down here. And this is just a big brown, just showing you the differences. And this is usually the view I look at, and I scan through, and I look for frequency and slope, and then I mark it down. So lots of fun work. This is just preliminary results that I finally finished looking at all of our data from last summer. So this is just looking at occupancy. Where we actually found them are the little squares in the red. So an interesting sort of divide here between the east and the west, but some of these areas up on the North Fork are extremely low canopy cover levels. Just, when you look at the aerial imagery of these areas, they're just little tiny patches of habitat. And even some of these squares in here, the places we're putting detectors are, along the edge of people's properties. I also included a lot of private lands here, so just going up to landowners that were in my grid square and asking if they minded if I put a detector on their property. So we're covering a really wide range of habitats. And one of the things that is really my next step is I want to break down each of these sites. I'm basically going to put buffers of different sizes around them and then analyze the habitat characteristics that way. So I want to look at habitat patch size, how isolated those patches are from other areas, the actual composition of the habitat, what type of forest and trees are there, how close that is to the coast, to other wetlands, a lot of variables that we're going to try to look at here. So that's really our next steps, is acquiring all those different variables and all the data for that. So that's how I'm addressing my first question, and I would like to continue doing that, as long as I possibly can. The DEC is really, interested in this as well, because it seems to be a great monitoring technique for this population down here. So, even after I graduate, I think they will still continue this effort as well. And the other thing I want to mention is I'm also looking at this same question on Nantucket and Martha's Vineyard. So really great study sites to go out and visit and spend your time at. But we are also seeing the same phenomena across those islands as well. So, Long Island is certainly unique and special, but I think there might be some other coastal areas where this is happening as well. So same study design, replicating everything as well there. So my next three questions I'm addressing mostly through netting and then radio tracking those individuals so I can locate where they're hibernating. So this has to happen in the fall time period. It's generally thought that bats enter hibernation by November 1st. That's kind of our cutoff date. But what we found last fall, and I'll show you, is that they were definitely active on the landscape way into November. So especially somewhere like Long Island, we might need to rethink these numbers that we've used. And then the other way I'm assessing this is by swabbing those individuals to detect the fungus. I can swab them in the fall, where you would expect that you'd have really low prevalence of the fungus. Unless they're going in and out of a site where they're picking it up, we really shouldn't be detecting it. And then I'm going to swab them again in the spring. Actually, in about 2 weeks, I'll be back down here and trying to catch individuals when we think they're emerging from their hibernation sites. And then the samples for genetics I can take anytime I catch a bat. So, what do? We string up mist nets. The net might be a little hard to see. It's not open here. It's just strung out across these two poles. Best places we have found to catch them are if you find a nice little rut in the trail where there's water, because they will swoop down and get a drink of water, or they find bugs to eat that are hanging out over that water source. So that's usually good to look for when you're going out netting. We started in last spring, kind of exploratory netting just to see, if we could detect the fungus on bats in mid-April. And we chose a site in Sag Harbor, Barcelona Neck, right on the state property. It's also a golf course. Really nice piece of land. And we chose it because we had really good detection acoustically the previous year. So just went out last April to see what we would get, and we got, I think, 5 individuals over 2 nights. Swabbed them all, and 4 out of the 5 were tested positive for white nose. So really interesting numbers in that we were detecting it. All the bats appeared healthy. Nobody had fungus growing on them. Their wings looked great. Usually you can see a lot of damage on the wing if they were really heavily infected that previous winter. So that was really interesting, and deciding to replicate that again this spring. But that's where we decided to go back and do our fall capture and tracking work. So this is taken there. at the Barcelona Neck property. So what did we do? We spent 73.5 net hours this fall, but that was really only across 5 total nights. So we really didn't put in a ton of effort in actually netting, and we ended up catching 6 individuals. 1 escaped, so we only were able to put tags on 5 of them. But we're hoping to get much higher numbers this fall. I'm probably just going to hang out and net every night for a month or so I can just keep putting out radio tags every night. And we were able to track them for about 14 to 23 days, depending on the individual. So the transmitters are pretty small. They're about 0.3 grams. You want to keep it small enough where it's less than 5% of the bat's body weight, so it doesn't impair their movements and their behaviors. But with that comes a drawback because the battery life is short. So these are only expected to last maybe 3 weeks to a month before the battery dies. We put them on with a little bit of surgical glue, so they will fall off as well after a certain amount of time. So we put these on. The other thing we can do is just take a swab to assess the degree of fungus, if there is any. Another way that you can actually detect the fungus, and it's been developed in the past few years, is by using UV light. So this was taken last spring, and you can just stretch the bat's wing over a UV light, and you see all these little fluorescent dots on the wing. It's not the actual fungus that you see fluorescing, but it's actually the lesions that the fungus has caused in the bat wing. So it's a really good way to see if the bat was infected recently, and you can actually measure the degree of infection by looking at the total wing area and seeing how much is actually covered. So a really good tool, non-invasive. The first method they were using was histopathology, so you'd have to kill the bat and look at the wing. But now we can swab and we can look at UV to get these numbers, which is great. So where did we find them? These might be a little dark, but tracking the bats involves a lot of work. So for the first, I think, 2 weeks, all of the 5 individuals we caught were just hanging out on the property at Barcelona Neck. They were all roosting in trees, not far from where we captured them. The actual property there is not very wide, so they're all hanging out in these areas, all roosting alone in their own tree, which is interesting. Usually in the summer, they form these maternity colonies, maybe anywhere from 5 to 40 individuals, but we don't really know that much about what they do in the fall. So it appears that their behavior changes. There's less of them roosting together in a tree. They weren't moving around very much, so they might switch a roost every few days, but they were picking a tree that was just a few feet away from their original tree. So at first, tracking was easy. We just go out every day and say, oh, the bats are just in this tree or in this tree. The other thing we would look at was do emergence counts. So it's really dark, but my coworker Mandy is laying here in the brush, and we're staring up at what we thought was a roost tree. Sometimes it's hard to pinpoint, exactly what tree your signal is coming from. Depending how the bat is sitting in the tree, if the antenna is sticking up, the signal can spray out like this, and you're getting detections all around you in a circle, so it can be difficult. But what we can do is do an emergence count at night to see how many bats emerge from the tree. So we never found any more than 1 bat coming out of a tree. We got some cool new technology to try this fall, these little thermal cameras that you can plug into your smartphone. So I'm going to try to use that and look at the roost tree and see if I can actually see the bat coming out. The other way to tell when it emerges, because it also gets pretty dark by the time they come out at sunset, is just to listen, have the radio on, and you'll hear the beep from the transmitter, and then once the beep goes away, your bat left the tree, even if you didn't see it. So it's kind of the best method that we have. But once the bats left the property, it was much more difficult to track. So she's holding a 3-element antenna here, so it's just an antenna attached to a radio receiver. And it picks up the signal. You just program it to what the frequency of your transmitter is, and you can pick them up. You just hear the little beeps. But once they left the property, we spent every day driving around with, holding the antenna out the window, just listening to white noise and waiting to hear a little beep. And you start going crazy and you hear all these phantom beeps. But when you actually hear a real beep, it's the most exhilarating thing. You found your bat, and then you can pinpoint down exactly where it is, where it came from. So I'll throw it out there. If anybody has free time in the fall, or students that would be interested, we could always use help tracking. We're going to try to stick to most of the South Fork again and stay pretty far east, but We might explore some other study areas as well still in Suffolk County, so let me know. So where did we find them once they left the property? This is a little northern hanging out under the trim board of a cedar shingle shed on Shelter Island. So this was the first bat that made a pretty large jump. It went from the Barcelona Neck property about 3 miles up to Shelter Island. We found it at Mishomick, hanging out in a tree. That was the first spot we found it. And then it went another few miles to this property. The landowner had died, and there was supposed to be a caretaker for the house, so the house was not in great condition. But we made friends with the neighbor, and they said, oh, just go over and look for the bat. So we found it hanging out on her shed, and it stayed on the shed until the transmitter died, so until about mid-November, when it was getting pretty cold out. So there he is. This is a view of what the shed looked like. It was pretty small, and it was just hanging out there. We couldn't always see it, but we could always hear it. So that was pretty cool. We did end up finding a hibernation site last fall. So this was roughly 6 miles from our original capture location. We had lost this bat after about 2 weeks, and we were kind of devastated. We couldn't find the signal for about 3 of our bats, and we ended up getting approved for flight time. So I didn't go up, but my supervisor and my coworker went up in the plane, and they flew around, and it's a really easy way to cover a lot of ground when you're trying to do tracking. And they were able to find this individual further east in East Hampton. Really nice house, really nice neighborhood. And here he was hanging out under the house in the crawl space. And he was there with another individual as well. So this was really cool and really exciting. The landowner is seasonal, so they don't live there in the winter. And she was totally enthusiastic and said, just go down to my crawl space whenever you want and check out the bats and do whatever you need to do. So we've had a lot of luck with some really great landowners that have been really accommodating and letting us go on their properties and monitor these bats. So what we did at this site was I put 2 acoustic detectors outside of the house so that we could see if they are actually coming out in the middle of the winter. We also put 1 detector inside the crawl space, so maybe see if they're just waking up and not emerging. And I just started going through all the data from the winter, and these guys are crazy. They were coming out on some nights when it was like 30 degrees. You wouldn't expect them to be out flying. Generally, like 50 degrees is your cutoff. So it definitely seems like they're doing a lot of interesting things. Perhaps this is why they're not being as affected by the disease. They're hibernating in really small numbers in very variable sites. Maybe they're able to come out on nice winter days and feed down here because you have this nice coastal climate that might allow for that. So these are all the kinds of things that I'm proposing and looking at here. This is just a picture of the entrance to that crawl space. So it's just a little grate, but there was nothing blocking this little area here. And just to throw it in, on Nantucket, we also have a hibernation site that bats have been using for 2 years. Also a crawl space under somebody's house, but it's very different from the crawl space we found here in the sense that there's a lot of light coming in. It's a little bit taller and just overall not what we thought we were going to find. So I think these guys are very adaptable and very unique, and this is a really interesting situation that we're seeing down here. And basically I think that this island and maybe a few others could really be a refugia area that could help the population survive and possibly combat the disease. So with that, I just want to thank all my collaborators. My awesome advisor at UAlbany, she's a disease ecologist studying anthrax, so she just jumped right into the bat world when I came at her with this project. My other nonprofit collaborators on the islands, so many great technicians that helped us. My technician that did all the acoustic work down here last summer just went out every single day, moving detectors, getting permission from private landowners. She was great. And Fish and Wildlife Service just gave us a really nice grant to continue this work for the next year, so I'm glad they recognize it's important. And with that, I will take any questions. Yes. Talk too much.
So, next Samantha at lunchtime?
Yes. And feel free to email me. Also, I'll just put it out there. If you have a crawl space under your house, I would love to check it out. Or if you know anybody that has a crawl space and you live out on the eastern end, I'd definitely like to take a look. And we have a nice survey that helps narrow down the conditions that we think we're looking for. So, yeah.
All right, thank you, Samantha.
You are welcome. Yeah.